My shoulders, knees, and toes, my bones, your music gets me through.
Hello, everyone. My name is Krych from the Mayo Clinic in Rochester, Minnesota. And on behalf of the ICRS, I would like to welcome everyone to the next hour where we're going to be discussing innovation driven by patient needs. It is my great honor and pleasure to share the stage with a true ICRS giant in Dr. Daniel Saris. I'm going to share my screen here for a few housekeeping items. So first of all, we had 217 registrants from 36 countries. So we're hoping that we have a very dynamic online session virtually here. The few things to note are, one is we do have active question and answers. So as you develop questions, please send them using the question and answer function at the bottom of Zoom. I will send those questions and ask Daniel Saris on your behalf. Second, this session is being recorded live, so it will be available to all participants. At the end of the session, the ICRS will send a link out so it'll be available to you. And then finally, you'll also get a survey, so we'd really like to hear your feedback about the session and the overall experience. The outline schedule for the next hour, I'll introduce Dr. Saris, and then he will give a 20 to 25-minute presentation on his perspectives of innovation driven by patient need. We'll then have lots of ample time for questions, answers, and discussion. So again, please formulate your questions as they arrive. Please send them in. I will make sure that they get answered in a timely fashion. And then towards the end of the hour session, I'll finish with a few announcements on behalf of the ICRS. So I would like to introduce Dr. Daniel Saris, a true cartilage giant and also past president of the ICRS. Professor Saris is a world-renowned orthopedic surgeon, clinician, and translational scientist with a focus on regenerative medicine and advanced knee reconstruction. Previously at the University of Utrecht, he is currently a professor of orthopedic surgery at the Mayo Clinic and recently became the first full professor of regenerative medicine at Mayo Clinic, an honor we are proud that an orthopedic surgeon was able to achieve. He has held many international leadership positions, including past president of our ICRS Society. He has expertise in every aspect of joint repair, from arthroplasty to sports injuries and cartilage restoration. He is the lead author on 2 of the most referenced and cited published RCTs on cell therapy, and was successful in helping register cell cartilage therapy in the EMA. The SUMMIT trial was certainly a key to paving the way for the only FDA-approved cartilage treatment in the last 20 years in the United States, demonstrating his impact on the field. As many of you know, he developed IMPACT when he was at the University of Utrecht, which is a cartilage restoration procedure where cartilage is recycled, mixed with MSCs, and implanted back into the knee in one surgical setting. He recently published his 5-year results in AJSM on the first 35 patients treated in the Netherlands. He is now running a successful clinical FDA phase 1 and 2 trial at the Mayo Clinic called the RECLAIM trial. Today, we are fortunate that he will share his thoughts on patient need driving innovation with all of us. I believe this provides us with his unique perspectives, namely in successfully navigating 2 regulatory pathways on 2 different continents. Without further delay, we have the privilege of Dr. Saris highlighting a few of those perspectives.
Thank you very much, Dr. Krych, Aaron, my colleague, my neighbor and friend, for that wonderful introduction. It's humbling, and it also shows you that my gray hairs aren't just from time. It's also been a lot of worries and troubles, but a wonderful adventure to share with a wonderful team. I hope this presents well. So innovation driven by patient needs. It allows me to talk about one of the hobbies, one of the passions that our team and I have had and have shared for many, many years. And when thinking about the topic that we were charged with on behalf of the ICRS, I thought the reclaim and the impact story would be a nice way of showing some of the thoughts that have gone into this process that we've been in for the last 10 years. These are my disclosures. So I've been fortunate, as Dr. Krych just mentioned, to have the benefit of working in a very fertile environment, and in an environment that was pushing what we call patient-centered progress, or innovation driven by patient needs, for a longer period of time. And it's been very interesting to work in this multidisciplinary field with basic scientists, clinicians, young students, patients in some of these societies very much involved, and also industry partners and financing bodies that would allow us or not allow us to do certain research projects. And I think that two of the big parallels between what I've noticed in Utrecht and what I've noticed here at Mayo Clinic is that there are some things that work really well in clinical care. And the trick is to figure out why they work well and improve on those going forward, but also use those examples of success to maybe solve some of the issues that are not as successful in other parts of our care. And the other side is that if you have a group of inquisitive people working together, they can also address what I think we should be calling still unmet medical needs. And there are still quite a few, especially in cartilage repair. So what we have in Utrecht has always been a team approach. And one of the things that I learned from one of our other past presidents, Bert Mandelbaum, was a team stands for together, everyone achieves more. And that's been very true in my experience. So all of these things that I will be presenting today and that you are aware of from literature or from previous presentations are all part of a team effort. And that's really important to say and to understand. We can't be on this stage with 12 giants. It would be a cacophony on Zoom. But it's clearly more than just me presenting my thoughts and my data. This is a process that we've been on with many people, influencing each other and pushing each other forward with a dual translational focus. And by that, I mean that I personally have always had more than just a clinical interest. I've been intrigued by biology and by science, and I'm probably genetically predisposed that way because of my parents, my grandparents, and the way I was schooled. But that also means that I, from an early age, had the idea that you can't just do science to do science. You also have to do science, I think, to solve things that are real in society, or in our situation, that are real in clinical care. So I think that some of the examples that I'll share with you show that we had a clinical reality and we had a scientific pathway to solve some of those dilemmas. And the other way around, we had clinical we had basic science information that was able to change something in clinical care, and you had to connect the dots or build the bridges. The other thing is that I feel strongly that it's fun, it's intriguing, and it makes a bigger difference to do first-in-man studies. So really doing something that hasn't been done before, it can either go wrong or it can make a big step forward and open up a field. And like we do very strongly here, do clinical research either based on procedures that have been done in the past and well documented going forward, or that are designed as very rigorously designed trials that answer a clinical decision-making process or dilemma that we have in clinical care that we need to solve to either be sure that we have evidence-based medical choices for our patients, or that we have sufficient evidence to show that the logistics and the economy of a certain treatment is worth all of that to the patient and to the payer. And then I also think that there are some examples where it is just important for us as clinicians to come up with ways to improve and simplify the current care. I don't think that there ever will be an orthopedic Nobel Prize because it wasn't given to the invention of the joint arthroplasty in time. And all the things we do will never be as big and as humongous as CRISPR-Cas or the other iPS cells and things close to cell therapy. But it's enough to just make a meaningful improvement on our care. And they can be small, just a change in surgical technique, just a change in diagnostic management, just a change in the way we approach a patient's care can make a meaningful difference. And I think we have a few examples of that I've been able to experience as part of the team. So early on in the ICRS, and I know you know these slides, but it's important to maybe catch on to some of the thinking that I have behind it. Early on, the people who really pioneered this field were confronted by naysayers and were confronted by what was called the complications of ACI. And the complications of ACI were documented as sidewall not integrating, as hypertrophy, as calcifications, as delamination. And all of these things were hindering first generation ACI. It is important to understand now, and many of us do, and some of us may not even be aware that this was an issue early on. It's important to understand that, in my opinion, these were not complications of ACI. They were just our misunderstanding of the biology that goes on in repairing a cartilage surface. And many of these had to do with timing of the procedure and the fact that we were not able at that point to use biomaterials covers or injectables. And we were using composites with injectable cells in a suspension, periosteal cover. Surgical technique was more burdensome. So for me, this slide is way more important because it shows that if you use the right technology at the right time in the right situation, Mother Nature will allow us to repair cartilage in an almost perfect way. And I know that doesn't happen every time in every single patient, but the fact that it is possible in biology means that we now have the challenge to make that happen more often and make that become the standard rather than the complications occurring. And we all know how we solved that. And those were really simple innovations. What I also took from it was this simple metaphor that I think is very important. And that is, fish will not thrive in a dirty aquarium. It's not fun to swim in a dirty pool. And as you know now, is that the environment around the chondrocytes that we were putting into these joints was not the same if you were treating a fresh defect or if you were treating an old defect, or if this was the third surgery that the patient had compared to if it was the very first solution. So very early on in the research that we started doing, I was intrigued by this idea of, can we maybe limit the complications of ACI? And can we understand the interarticular environment? Because I was quite sure that the basic idea that Lars and Mats and the others, Dan, came up with of using cultured chondrocytes was a good idea. But I thought we were missing something in the external environment. And that was the time when in Germany, the Orthokind product was on the market, and we started doing research with this. And in no way is this me saying that you should use Orthokind. It's also not me saying you shouldn't. It's just showing that we had this idea of the joint needing to be treated either before surgery or between the biopsy and the transplant or after the transplant to optimize the environment, to clean the tank, to prepare the joint for biological reconstruction. And this is one of the first examples of an orthobiologics therapy trying to influence the interarticular environment. And with a bunch of really good friends and clinicians, we were able to do a prospectively randomized trial independent of industry that was very conflicting in its outcome, because we had to report that it wasn't statistically significantly better using the Orthokind preparation versus a saline placebo, even though we did see that in the co-symptoms, in the sports, in some of the quality of life measures, people clearly had an improvement. But we didn't understand trial design well enough, or at least I didn't at that time, because we had designed the trial to measure the patient's outcome for pain as the primary outcome measure. And at 1 year, the patients reported similar scores in pain, even though the Orthokind was always a little bit numerically higher. It wasn't statistically significantly different. So in the official publication, you will see that we say, well, it wasn't statistically significantly better because we had to say so. But it did spark my intrigue because clearly these patients were doing much more. Their joints were feeling okay, and they were able to have a better quality of life in sports and physical activities. And when we looked at them again more recently, at 10 years, we could see that even though many of these patients didn't even remember which of the two treatment groups they had been in, many of these patients who had been given the real component had not had any form of subsequent surgery. So to me, this difference in the curve here clearly shows that there is a disease modifying effect to some extent, because after 10 years or after 5 years, you can't just say it's a placebo effect. There has to be something. So I still think that this is applicable to some of the orthobiologics that we are trying now. And it still means that there is an unmet scientific need, which is for us to better understand how orthobiologics can be applied to improving the outcome of cartilage repair, of joint preservation. And that is something that I would put on our to-do list as ICRS members. It's also clearly not solved from a prospective cohort perspective, because as you saw from the publication that we put out last year, when we use the more modern preparations that are available commercially now, which we felt did very well in clinical care, and the patients and the logistics are fine. Unfortunately, in this group of 140 patients that we treated in the Netherlands, we weren't really able to show a clinical beneficial effect. And that makes me think, so we're 10 years further, we still haven't managed to understand it. And there are many of these products on the market now, many different aspects that we're trying to understand. And that is still a challenge. I think we need to make a concerted effort with other societies, with multidisciplinary groups to figure this one out, because I truly believe this is the next hurdle to our improving the outcome of joint preservation. So what have we learned from this so far? I feel that we've learned that patients will be more active up to a certain level of discomfort. They will accept that level of discomfort and just do more. So just measuring pain is not enough. And you saw that in the Summit trial, where we combined pain and function as outcome parameters, which is different than we did in the Chondro Select trial or in some other studies. That means that we need to think well about trial design and about endpoints to come up with the right answers to the questions that we're asking, and not have to negotiate around the clinical reality, even though we will always be surprised by the data and we will just have to present the data as they come. Thinking about it and learning from previous experience is one of the big things that I think has driven patient care in the right direction. I also think that there are still quite a lot of confusing pieces of evidence out there. You see that in Europe, we had a couple of different cell therapy products on the market. And at the moment, to the best of my knowledge, for cartilage repair, there's only one. The two that we trialed and were involved in were only commercially available for a very short period of time, and for whatever commercial, strategical, or regulatory reasons have not made it to become part of our standard package of care. And I feel regret about that. And it also creates sort of a moral dilemma, because how are you going to ask your next patient to randomize in a therapy of which you're not certain that it will really change clinical care 5 or 10 years from now? And I think we have a shared responsibility to work on that. I also feel that we need to be both open-minded and strategically smart with innovation and testing new products. And I think that some of the confusion and some of the inconsistency in data that there is on HA, on viscosupplementation as a wider field, but also on the biologicals such as BMAC or MSC injections or platelet-rich plasma, PRP, and some of the others, the amniotic fluids, are probably something we can manage better. I think that the fact that industry and investigators come up with all these new preparations and new solutions are perfectly logical, make sense, and push the field forward. But the moment that the data sets are small and the products, for instance, as in HA and maybe also a little bit in PRP, are presented as competitors, and so my PRP is better than yours and my leukocyte rich is better than your leukocyte poor, it sort of takes away from the overall point of view that we're all doing this to come up with a reasonable explanation of the science behind this product, beneficial to the patient's clinical needs. And I think we can do better there. And I think there are also examples of that to some extent. So not veering off the topic too much, but if you look, for instance, at another part of science in biology, the whole field of CRISPR-Cas was very rapidly evolving, very interesting scientifically, very well basic science established. But many of these groups were aggressively competing with each other. There were patent wars. There were people being bought from different labs. And if you read some of the popular literature about this, this was clearly a field that was trying to find its way, developing rapidly, but also in sort of a competition mode. Until COVID came along, a few people stepped up and said, this is such a large problem. This is such a large unmet need that threatens mankind. We need to bundle our energy and focus. And that's what happened. And within a few months of 2020, the diagnostic testing, the vaccine development, and some of the real solid basic science that was there now transformed into a collaborative effort, which is worldwide, which was combined with novel ways of open source publishing, putting out data sequences, sharing raw data, that really has accelerated this field and has also been acknowledged by the awardants of the Nobel Prize last year to Jennifer Doudna and Emmanuel Charpentier. So obviously cartilage repair doesn't combine or doesn't even compare to this closely, but it did spark to me the idea that one of the things that we as the ICRS and as a field of biosurgical scientists and clinicians and basic scientists can do is try to focus on things that we can collaborate on and make a bigger difference than this trying to push the cart by ourselves. An example in our field, for instance, is there could be a benefit from maybe sharing trial data and sharing protocols upfront or as we go, rather than this being a company protected piece of information. And I think this is a very nice example. Good colleagues and friends of ours worked hard on putting together a very, very robust trial design. And then the trial design probably in the end was so robust that a good technology that was clinically performing quite well and not necessarily better or worse than some of the things that are on the market now, did not make it through the process here in the United States for FDA clearance, just because of the original threshold that was set, and that was very high bar to pass. So one of the things that I've learned from this, and that I would advocate, and we could maybe even do in our journal or in the new journal that Tissue Engineering has opened, is publish study protocols. If you think this is a good protocol and you want to share the protocol, publish study protocols so different groups don't have to reinvent the wheel, and we can learn from each other in an early phase, like we were able to learn from the trials that Dr. Krych mentioned in the beginning. And the trials that we did with the Belgian group and the people in Poland and the Czech Republic were instrumental in our understanding of how to run our clinical practice in a way that is GCP compliant and gives you good data, but also in a way that allowed us to do the regulatory trials to get new stuff on the market. And that is really important. The other thing that's really important is to remember that cartilage repair has been here for more than 30 years from a cell-based perspective. And as you can see with advanced imaging, cartilage repair, if done well, is usually successful. It doesn't have the complications that we documented before, but it's come a long way. It's come from cultured, injected, uncertain which kind of cells they are under a periosteal flap, to now one-stage injectables, very reliable pharma-level treatments. And I don't know what a good comparison is, and I definitely don't want to compare the first generation cartilage repair to a horse and carriage, and I don't think that what we have now is a red Ferrari. But I do think that the metaphor some point does play an important role, because you wouldn't expect current patients and current providers to still want to use technology that was there 30 years ago. You want to learn from what you see in clinical practice, what the patient tells you that they're looking for, to make sure that you come up with little incremental innovations or big change. You also want to accept the reality that in some societies, it is not possible to have a high-tech bio solution for every single patient. So we also have to have off-the-shelf, simple fixes, maybe biomaterials-based, maybe cell-based to some extent. And then there are actually situations in which a more basic, well-reliable workhorse is better than the next level sports car from cartilage repair high-tech labs. So that's what, in my thinking, we always try to do. We try to fit the patient's clinical situation with our understanding of their needs, and then figure out which of the things in our portfolio best fits their needs. I don't think there is a single best treatment, and I also don't think that microfracture is the golden standard or the comparator for trials. So how does that translate to what we've been trying to do in cell-based cartilage repair? Well, I think that the understanding of the human genome and now being able to do genetic testing on the cartilage cells to improve the quality of our culture process was really important. The fact that the Genzyme and then Sanofi and now the Vericell company have developed release criteria and potency assays and assays to make sure that what we put in our patient has a good quality, a reliable consistency even though it is made from the patient's own cells, is a next level quality assurance and a safety standard that we would expect from our drinking water or from our vegetables or meat. We would expect it from chemicals that we use in other parts of medicine, but it wasn't there in cell-based cartilage repair, and it is now. And those are important big change innovations that have made the outcome of therapy much more reliable. It also made us as a group think, and this is where Lucienne and Jorgis and Tommy and all the people that you know from literature and from our group being on stage got to thinking, and we said, you know, we can keep on going with 2 stages and 1 cell type, but that might not be to the best of our understanding of joint homeostasis and patient's needs. We might be able to switch to 1 stage using 2 cell types and some way to manipulate the aquarium and to clean the joint. And as you will see in the subsequent webinar, there are also groups trying to work with 1 stage, 1 cell, which might be a scientific challenge. I'll be up to that discussion anytime we need to have that discussion. But from this part, I think the lessons learned are it's hard to move to hit a moving target with an RCT, and regulatory bodies were changing the rules for some of these trials, so it was hard to perform. Microfracture is not the golden standard. So as far as I'm concerned, in the future, that should not be the comparator for trials. We have to come up with more innovative trial designs and innovative statistical ways of figuring out what the reality that we can offer our patient in the future is. And I think it's really important for all of us to really embrace the concept of good clinical practice, good manufacturing practice, and regulatory oversight. They are our friends. They are not the devil. And if you are afraid to live up to these, there's probably something wrong with the way you're thinking about what we can really do for patients, although the administrative burden is considerable. So how does that all translate into impact, into reclaim? Well, I think you know where this is going. So if we agree that the environment is important on what the cells do, then it is important that we now know that MSCs have an influence on modulation of the immune system and on the secretion of trophic factors. This has been shown in clinical patients with osteoarthritis, where the amount of MSCs goes up with the grade of osteoarthritis. The amount of MSCs goes up with the amount of cartilage damage after ACL repair. And to me, this meant that Mother Nature, the body, is trying to heal something in the joint by using MSCs. And that's an important signal. It's not something done by cartilage by itself. MSCs play a role from Mother Nature's overall grand design, but it comes up short. We now know that we don't call them stem cells necessarily. We call them medicinal signaling cells, and maybe the names will change again, but it has changed our concept of thinking what these cells do. Maybe these cells don't necessarily make tissue, but provide factors for the immune system to respond appropriately and tissue to be formed in the most effective manner in the clinical situation. And then doing an arthroscopy in a cartilage repair patient, it always intrigued me why these cartilage flaps were mechanically insufficient, but biologically still viable. So from an early phase in my career, I always was intrigued by finding a way to repurpose these pieces of cartilage. And that's basically what Reclaim, what Impact does. So in any cartilage repair procedure, in the final treatment, you debride the defect, you clean the defect, make sure you have stable, nice new walls to work with. That tissue went to the lab for research, and now it goes to the lab for repurposing. We mince it, we chemically digest it to release the chondrons from that tissue, and we mix those chondrons with allogeneic, previously cultured MSCs from the donor bank. We can put those into an injectable solution because fibrin glue is allowed to be used in the joint. We do that often as part of the Macy procedure or previous cartilage repair strategies. And now we can use it as a carrier vehicle, which takes care of the cutting and pasting and the suturing, and in our hands has been shown to be quite reliable, which now allows us to be more effective clinically. It has been shown to be safe, and it is quicker and cheaper than currently available measures. So in the trials, both in Europe and now in the trial here at Mayo Clinic in Rochester, we still do this in an open procedure because we want to make sure from a regulatory perspective, we don't have the innovation of arthroscopic development and the surgical treatment and the cell biology interfere too much. So at the moment, we use an open procedure where we use the cookie cutters to size and shape the defect to make sure we don't take away too much. We have stable rims. And this allows us in a very reliable, quick and safe way, both for the MSC patients in our standard of care and in the reclaim, the impact patients in our studies to harvest the tissue that is mechanically insufficient for the patient to not have clinical symptoms, but still has the biological potential to provide us with a good source of cells that will heal the defect if we provide the right environment. So this is an example of a femoral condyle in a different patient where the final stage of the procedure is done. And really, this is not rocket science anymore. And we can do this in an arthroscopic procedure for the knee. And after the trial is completed, this will be the next innovation, because then we don't have to do the arthrotomy anymore. The fibrin glue sets, it has the cells in it. And we know now from the 35 patients with 5-year follow-up and with the first group of patients that we're following up here at Mayo that these results are safe and they're clinically really intriguing. So the safety profile, as expected, isn't a big issue. We have not seen any safety issues, no adverse events or serious adverse events in the 35 patients in Europe for 18 months and now 5 years, nor in the first group of patients here at Mayo that we still have to report, and we will hope to report on the first group of 1-year results here soon. We also now know at 5 years, presented that the last time we really saw each other face-to-face in Vancouver, that the 5-year outcome clinically is really interesting and, in my opinion, similar to the outcomes that we saw with MSC, that we saw with ChondroSelect, and that we see in prospectively published real-world data cohorts. These patients go back to an active lifestyle without too much knee pain after 1 surgery using cell-based repair, not scar healing. And then I think it's really interesting to see the serendipity and the opportunity of the scientific developments while you're doing this step-by-step innovation, because not only were we able to show that you can do cartilage repair with cells in 1 procedure, we were also able to show that histologically, the tissue designs the various layers the way the tissue needs to divide those layers, and it's different than scar tissue. Even though you will not hear me call it hyaline normal cartilage, it is well-organized tissue with all the functions restored to some extent. But also, we're able to benefit from developing technologies by which we could now follow if the MSCs has made the tissue or if the cartilage cells has made the tissue. And I still remember us talking about that as a group. How are we going to trace these cells? Because 8 years ago, the technologies that we have now were not available. And we can address that a little bit later in the Q&A. But the basic lesson was you need 10% of the patient's own chondrocytes or chondrons, maybe 20%. And you mix those with allogeneic MSCs, and after a year, most, if not all, of the defects had healed, and you can only find the patient's own cells filling the defect. The allogeneic MSCs are gone. And that brings it back to the dual translational role. So these slides are hot off the press. This is work that Jasmijn Kurpershoek and Lucienne Vonk are doing back in Europe at this moment, because we're still intrigued by how do these cells communicate, and do you really need 2 cells, and do they do something together, or do they do their separate tasks? So these images and some of the active tracking that we've been able to do actually show that there's mitochondrial transfer of information from the MSCs to the chondrocytes. There's also a little bit of backtalk from the chondrocytes to the MSCs, and we're still trying to figure out and go into the details. And I look forward to Jasmijn's PhD and her being on stage presenting this wonderful work that she's been doing. But I wanted to be able to use this webinar as the first opportunity to share some of that data that shows that if you look at the communication the way we did between chondrocytes and chondrocytes, there's some, but it's not major. If you look at the communication between the MSCs towards the chondrocyte, there's clearly an uptake. It's quick, and it's clearly more than when you look at the communication between the chondrocytes towards the MSCs or the chondrocytes to the chondrocytes. Does that mean we understand what's going on? No, but it ties into the data from Sakaya that showed that MSCs go into the joint to try to fix if there's a cartilage problem. It makes us understand why using allogeneic MSCs might be better or even just as good as using cultured chondrocytes. It helps to solve the interarticular environment. And that means that we now here at Mayo and in Utrecht think about this as a platform technology where we use tissue recycling with or without MSCs to address injection options for early osteoarthritis in the hip and in the knee. It allows us to do a trial in Europe and a trial here in America, which are slightly different, to validate this technology in 2 different institutions, to further develop this technology, to be able to use it for the hip, to be able to use it for meniscus repair or to augment meniscus transplantation, and obviously also to work on osteochondral solutions that can help improve the results of the already great findings that we have from allograft transplantation. So finally, where does this drive us, and how is this patient-centered progress run by my group here with Dr. Krych and Dr. Kusters and Dr. Vonk and the group back home in the Netherlands? Well, obviously in Europe, we're working on the 10-year follow-up. I think long-term data are important. They are doing an RCT with impact versus standard of care or Mother Nature. And we're trying to work with Dr. Carperin, Professor Carperin and his group in Twente on measuring joint homeostasis and advanced imaging to better understand how to modulate intra-articular homeostasis with these bioactive agents that we have. While meanwhile, here at Mayo, we're validating the reclaim therapy in a new group of patients with adipose-derived MSCs, and we're developing the technology for other joints and other parts of the joints that are important for us to learn and how to treat as a joint preservation clinical basic science group. So thank you very much for sharing this time and this initial talk, and I look forward to the interactive discussion, and I hope we've left enough time and sparked some interest for us to continue in the webinar with an interactive debate.
Well, thank you very much, Daniel. That was absolutely outstanding. As we're on the theme of giants with ICRS, we're fortunate to have a few giants in the participants. I see a lot of friends and familiar names, and we wish we were seeing all your faces, but we do have Daniel Grande, Susan Chubinskaya, Arnold Kaplan as in the audience, and I would like to invite them, if they don't feel too much put on the spot, to become a participant, and if they had any questions or would like to have any discussion, but I'll work on inviting you as a participant. We have a few questions that are coming in. One that relates to bone marrow stimulation. So we saw you said it's important to publish protocols, and notice that your control is a non-operative treatment. Obviously, previous trials have compared to microfractures, the gold standard. The question is, where are we on marrow stimulation? Would next generation bone marrow stimulation be a suitable surgical option or possible comparator in some of these trials?
Yeah, I think that's a very relevant point to address, and I think that the development of next generation technologies for bone marrow stimulation are very important. Like I said earlier, I don't think there's one solution that outperforms any of the others. And even though I don't think that microfracture is the gold standard anymore, or maybe never was, it was a comparator that we needed to use as a stepstone in trying to improve our therapeutical options. Do I think that calling it micro drilling or nano drilling, or do I think that adding a membrane really improves the outcome and changes the clinical course? I don't know. Have I seen good data that shows that it makes sense to consider it? Yes, for sure. So just as much as on the cell-based part, there needs to be development. Hi, Arnold, welcome. There definitely needs to be development on the Mother Nature's natural repair response. But if you feel that cells from bone marrow stimulation play a role, then I think looking at the work that Alberto Gobi and others have published, where they use bone marrow concentrate without the marrow stimulation, might be beneficial, because it may get us away from some of the negative effects that we've seen from microfracture in the earlier data sets. So I want to be open-minded to that.
So Daniel, great talk as usual. And of course, you're wonderful for not calling the MSC stem cells, and I applaud you for that. We are working on ways in which we can try to change the lineage of MSCs into functional articular chondrocytes, and we have RNA-seq signatures of real chondrocytes, and MSCs. So we have targets for trying to get those cells into the lineage. What's crystal clear from our work is that all of the marrow MSCs become hypertrophic if they become chondrocytes. And so that's a destructive event. And this business of communication is the black box by which we basic scientists have to do better in understanding exactly what the MSCs are putting out. And I would caution you that, of course, the Kaplan dogma is that all MSCs are MSCs, whether they're from fat or from marrow or from wherever, except when you do single cell RNA-seq, although they have 17,000 transcripts in common, they have about 1,500 transcripts which are unique to the tissue of origin.
Yep.
And so the question that I always get that I can never answer, which you'll provide data by these clinical trials, is which is better? Which is the one which is the most receptive to that knee joint microenvironment and puts out the right spectrum of factors? And I think that's the black box that we basic scientists have to do a better job in trying to understand.
Yeah, Arnold, thank you very much for adding that and for sharing the data that we were sort of waiting for. So that's really interesting to know. I'm going to go out on a limb and say that, and I've joked about this before, but I really believe it's true. So cells are smarter than surgeons. I also think that cells are smarter than scientists. And that's why I like the idea of using the MSC rather than the extracellular vesicles or rather than the mitochondrial end products, because I think we will probably make the same mistake as early on we did with BMP. BMP works in a lab, but it hasn't shown a big difference in clinic yet because we don't understand everything that goes into that process. And the cells can sense, they can respond, they can adapt, and they do so in a different way. And that's why we think that it doesn't make a difference if you use your or mine MSCs because they're going to do their job anyway. I personally think it's a benefit to use allogeneic because they will not form tissue and we will not become confused. And I also think that if the MSCs can help cartilage make cartilage, that's always going to be better than if we still try to have MSCs make cartilage for us, because that's not what Mother Nature designed MSCs for.
So fantastic. Before we get to Dr. Grandi, so there's 3 questions that have come in on the topic of MSCs. So hopefully, Dr. Kaplan, you can stay with us for this. So question number 1 for you, Daniel, is cell delivery. These MSCs, can they just be injected into the joint, or do they need to be put in a local scaffold into the defect? Question number 2 is what's better, adipose versus bone marrow derived MSCs? And then question number 3, you kind of hit it on allogeneic, is one donor or multiple donor pool for the MSCs, if you guys want to take those 3 questions.
Yeah, so the first one is we're doing trials with MSCs for osteoarthritis as an injection therapy, and those are just injected into the joint to modulate the interarticular environment. I think that if we look at that as an OA treatment, is a whole different story. My story now is on treating cartilage defects, and I feel that cartilage defects need to be treated by putting the cells in the defect, and therefore I feel they need to be surgically administered. And this can be done with a scope or a micro or nanoscope. They can be as minimally invasive as possible, but we do need to put the cells where the disease is, which is in the defect. So that would be my first answer to that. I hope that we are able to show that the trial in Europe with bone marrow derived MSCs and the trial here in America with adipose derived MSCs will not show a clinical difference. I think that there are reasons biologically to expect that they will be the same, even though I think that it is easier to harvest adipose derived MSCs, the yield is higher, and therefore the culture process may be a little bit more simple. From a lab and a GMP perspective, there should not be a difference. And then the third part, I think that allogeneic is better because it is easier to do in a one-stage procedure. You cannot do autologous in a one-stage procedure, which again puts up the cost, puts up the logistics. And we don't know if you need one donor or multiple donors. Sort of a fun story is that initially Lucienne and Tommy and I designed the technology where all the male patients were given female MSCs and all the female patients were given male MSCs, because then at least we would be able to do XY testing to see which patient had which tissue grown from where. Otherwise, we'd have to do whole genome testing, which 10 years ago wasn't that easy. And meanwhile, as the trial was ongoing, the people in the stem cell lab in Utrecht said, you know what, the bone marrow transplant group has come up with this short triplicate testing. So you only have to do 6 or 9 pieces of DNA to know if it's yours or mine or somebody else's. And that's why we ended up using the STR and not using the XY chromosome. But we designed the science to go one direction, and then evolutions in the rest of our field helped us solve a question that was scientifically really interesting in a totally different way. So I think that a few years from now, we will have a pool of donor MSCs, which we can now relate to the interarticular environment that we've learned how to measure better. And then you can say, you know what, this patient's chondrocytes need an MSC with this or this or this characteristics, just like some people prefer ketchup or mayo or mustard on their sandwich. You can title this to the patient's needs.
Excellent. Dr. Grandi, I give you the floor.
Oh, thank you. And Dan, great.
Hi, Dan.
Talk.
Good morning.
How you doing? Good to see you, and congratulations on all your excellent work. And I think you actually asked I had 2 quick questions. One was, did you, and I think you've answered it, and that is with regards to the MSCs, priming them prior to cultivating them in a condition that might be more anti-inflammatory or might be something that I think you've alluded to that in your response. But the other question I had was with regards to the morselization or the mincing of the cartilage, is there a specific size that you look for, and is it automated or do you do it by hand? How is that accomplished?
Yeah, thank you very much, Dan, and great to see you again as well. Obviously, I see some of your Facebook posts with the wonderful boating trips that you guys do. It makes me sometimes jealous. Hey, so I think one of the things you just mentioned there is really interesting. So like I said before, the cells are smart. So maybe the best sensing mechanism is the cell. So what you propose is something that we've been thinking about, but we haven't really done yet, which is take synovial fluid from the joint of a patient, put it in culture with the MSCs, and then use those MSCs that now have been exposed to the environment of the patient's joint and mix them with the chondrocytes in the procedure. Because now they are aware of the situation before. It may work, it may not, it may be an unnecessary complication. It's scientifically super interesting. And I do think that it will help us answer some of these questions on what are the factors in the joint that the MSCs can help to improve, both in a modulatory of immune effect, but also in a trophic effect. So that's interesting, but we don't have it solved yet. And I don't think others have either. From a mincing perspective, it's interesting and we're still learning, but I think, and I'd love Dr. Krych to pitch in here, I think what we've learned now is that if the tissue is still pretty healthy and mechanically solid, it's much more difficult to get a good yield of high-performing chondrons from it. If the tissue is a little bit more fallen apart and fibrotic, it's easier to chop it up and to chemically digest it, and you have a higher yield of cells. So that's one biologically important aspect. The other thing is that we are still working on mincers. So there were commercially available mincers that seemed to do an okay job, but didn't really make a big difference once we've done the chemical release, and they were too expensive. There were less expensive mincers that didn't have as much durability. Some colleagues of ours, and Dr. Krych also, have been working on situations where we use shavers in the normal arthroscopic procedure to capture the tissue that you now harvest with a shaver and then run it through different sizes of shavers with different blades to morselize the tissue. And the smaller the tissue fragments are, the better the yield after chemical digestion. But we don't have numbers that we always aim for. We're still developing the release criteria. Aaron, anything you want to add on the yield from your perspective in the hip, the stuff that you've been doing there as well?
Well, I think you have to keep in mind that we're constrained by the time of surgery. The patient is literally, under anesthesia. So Daniel and his team in Utrecht developed this rapid, protocol, but ultimately we're constrained, by that amount of time. And I agree with everything you said. I think somewhat damaged, more surface area. I think we do get a higher chondron yield in that cartilage. One question from the audience, Daniel, is, does that higher yield necessarily mean better patient outcomes or better, findings on the postoperative MRIs? Do you have any observations there?
Yeah, we don't know that. So we, from the Dutch group, we were able to correlate yield, and we did high and low yield, and the clinical outcomes were comparable. In the current study, we don't have that data released yet, so it's still under investigation. And I would venture to say that at least if we know that we reach the cell numbers that we aim for, which is 2 million cells per square centimeter, like in ACI, then we know that we get reliable, good results in the clinical outcomes and the tissue fill. But I don't think we have the dosing figured out yet. And that's also because we're just doing phase 1 studies. I think
Daniel, I would say one thing about the chondrons, which is if you have an intact cartilage, those chondrocytes are set up in a maintenance metabolic state. If they're disrupted in a big tangle, they're activated, and those are the activated ones that you're most interested in, and I think that's the answer. So when we use digestive enzymes, you can't get out the intact chondrons. Very difficult to get them into suspension, and so that's the yield issue. So again, your surgical observation is matched by our ability to characterize the chondrocytes.
Oh, that's really interesting addition to that information. And that would then also be a challenge for groups that are using within one procedure minced cartilage without MSCs, which is what the ICRS will be talking about in the next webinar a few weeks from now. Aaron, I think there's an interesting question here by Diana. Did you see that one? She asks, and I think I remember her because she's one of the students from the Netherlands we had a presentation with not too long ago. It's really interesting because Diana asks, how do we get the patient's needs front and center, and how do we power these translational studies and make sure that we as clinicians and as scientists are really aware of the patient's needs, and is there a need for needs studies and needs assessment in driving these innovations? And my answer would be big yes. So anyway, we can figure out what the patient's needs are, what the unmet medical needs are, technology, patient vocal groups, developments there, very much in favor of them.
Yeah, I think that's what you have to keep our eyes on. We all design these trials because we want to see, statistical differences, but the question is, does that mean meaningful improvement to the patient's life? So I think that's a very good point. Couple of MSC related questions yet. So one is, do we know if there's any role for what you're doing in terms of the allogeneic MSCs versus the possible autogenous MSCs that are recruited to the site just by the surgical trauma, et cetera?
Yeah, I think that's a very interesting point. I would say that obviously they are being recruited, as we saw in the work from Sakaya, and that's still going to be similar. But I don't know how, because we haven't measured that. I wouldn't know how to measure that at the moment. One could argue that that process is going to be complementary to what's already happening. One could also argue that it might be downregulated because now there's a high dose of allogeneic MSCs, and there may still be other options that people can hypothesize. So yes, I think that is a factor, and I think it will always be a beneficial factor. I don't think it will be a negative, but we need to learn more about that yet.
And then our friend James Sweet asks, is there a role for that post-operative, control of the homeostasis of the joint environment? Are you started your presentation with PRP and Orthokind. Is there a role for controlling that homeostasis post-operatively in the knee, or do you just see a difference by adding stem cells or MSCs to your chondrons?
Yes. And thanks. Good evening, James. Great to hear from you again. You're always online with ICRS events and ESCA and everywhere, which is wonderful. So very early on, initially, I thought, you know what? Why don't we use the time between the biopsy and the ACI to optimize the intra-articular environment? And that's where we thought of using Orthokind or PRP or something else in those weeks in between. Obviously, there's people using low-dose corticosteroids or HA or PRP after regenerative joint preserving procedures at the moment, but I still think we're just doing potluck research there. And my answer would be yes. I feel that treating the joint appropriately after a regenerative procedure will influence the patient's rehab and outcomes and improve the outcome of the tissue repair. And I'm going to ask you a question, Aaron, which we didn't discuss up front yet, but have you noticed a difference? You've started seeing reclaim patients and how they do early after surgery and how they do the first few months after surgery. And how does that compare to the MSC patients or the other patients that you've seen? Do you see a difference, or are they the same?
Well, I would say, you know, for all surgeons that do cartilage surgery, I mean, there is a huge spectrum of, you know, recovery and rehabilitation. But I would say overall as a whole, I am pleasantly surprised that these patients, you know, with reclaim, I think do have less, you know, postoperative inflammation, less postoperative swelling. I do think they get their muscle strength back a little bit quicker than, you know, just the gold standard cartilage procedures. I've been pleasantly surprised by that. I would also say that the 6 month, the 12 month, the 18-month MRIs at this point have become very predictable. You know, I think with some other technologies, it's, you know, things don't always look as you want them to, but I think in the patients we've treated, it's just, you know, it's wonderful to see that maturation of tissue over time. And I think it's become very predictable in terms of what to expect when you're getting those MRIs. So I've been pleasantly surprised.
Yeah, and I think that's also what Roel Kusters and I saw in the patients in Utrecht. And obviously, I'm biased because I like the therapy. That doesn't mean I'm wrong. But it could be that the MSCs who are in the joint after surgery help the post-operative recovery of the joint, which is what James offered as a suggestion. So lots of work to do. And I think we're out of time. So I'm handing it back to you, Dr. Krych.
Yeah, and that really answers our last question from Felix about, you know, we are looking at structural, you know, MRIs to follow the, you know, the cartilage regenerate. So I'll just share my screen here for some final announcements. So thank you everyone for, you know, really a wonderful and dynamic session, even spontaneously putting Dr. Grande and Kaplan on the spot. Please join us for the next ICR webinar on the 7th of July. Christoph Elser, Mats Brittberg, and Gian Salzman will talk about cartilage regeneration for Mintz cartilage repairs. And then finally, continue to submit your work to the Journal of Cartilage and Joint Preservation. It's only been 50 days from submission to acceptance on average. So thanks for your support. Thank you to the ICRS. Thank you for an outstanding session. Hope everyone has a great day.
Thanks, Aaron. Thanks, ICRS. And thanks, friends. See you soon face to face.